session of the meeting. And the topic for the remainder of the afternoon is discussing resources and services for advancing drug discovery, something that is not as glamorous as many of the previous sessions, but a topic nevertheless extremely important, and I hope you find the information in this session useful to you. Now, if you're doing drug discovery and you're serious about it, you're in it for the long haul, sooner or later you will need to outsource some of your work. There are a lot of services out there provided by contract research organizations, academic drug discovery centers, the NIH. And navigating through this labyrinth of help is no small task. So we will begin with Dr. Marcy Glicksman. She will give us an academic perspective on drug discovery services, both centers and CROs.
Good afternoon. First to thank Howard for inviting me to participate. This, I think, is my 5th year, and even though I feel like I've been doing, I've been doing drug discovery for now more than 20 years, but I feel like actually being at this meeting, I'm always learning new things. So I think it's been a great group of people and speakers so far. So what I'm going to tell you about is just sort of quickly give you an overview of the kind of resources that are out there for information, as well as resources in terms of how to pick CROs and where to go for various places. And then, my own perspective in what we have used at our drug discovery center. So first, why outsource? And then I'll talk about various kinds of academic resources, and then I'll end with contract research organizations. And just as a disclaimer, there's no way that I can be complete. There has been some mention from some of the speakers of some CROs. So I'll just give some examples. But there are directories out there, and a lot of the best resources are your own colleagues, finding people that have had good experiences, and starting there. So first, why outsource? Basically, it's as simple as that there's a lack of internal resources. There's nobody that can be an expert in everything or have everything in one place. And so really, that's when you look to outsource. Also, lack of expertise. So even though we like to be generalists, or at times like to try to be generalists, it's often good to get experts. You have your own expertise, but then to find people who have other expertise that complement you. Also, lack of proper equipment. Sometimes you have certain needs, but you actually don't have the right equipment to do it, or within where you are. So that could be scaling up for chemistry. That could be robotics. If you want to do a high throughput screen, it could be chemical libraries. So those are other things you look to potentially outsource. And then flexibility. So a lot of times, like for example, at our center, a lot of our ADME work we outsource because we just don't have the needs. We could develop those assays and have them in-house, but we don't have enough of a volume to really make it worthwhile. So it gives you some flexibility to be able to outsource specific things that, that you need at the time. So, and this is just a reminder of how many different aspects of drug discovery are required, and what kind of resources you need for each of these phases. And so I've listed, for example, in discovery itself, that you may need bioinformatics, you may need target ID help, target validation, assay development, virtual screening, etc. So all of these things just for discovery. And then if you go on to preclinical, again, there's a long list of resources that you need for this phase. And then, of course, clinical, and then the approval. So again, just to reiterate that this is all good reasons why to look for where you can get resources as well as expertise elsewhere. So then in terms of what to outsource, I mean, it's a huge industry, And basically every segment that I showed you previously in the drug discovery process can be outsourced. And in terms of what to decide, what to outsource, what are your core competencies and how can you find people that have expertise in areas outside of your own expertise. And then also in terms of what resources you have. So you may not have resources to really do scale up chemistry, for example. And then there's certain aspects of drug discovery in which there's sort of gold standards that exist. For example, CHAN test has been one of the gold standards, for example, for measuring hERG channel activity. There's other CROs that offer binding type of assays, which is a good first pass, but in order to file an IND, you will have to have the kind of assay that CHAN test offers. Also, preclinical toxicity studies, there's certain requirements and certain standards that have to be followed. Also, GLP and GMP, these are all very detailed, and there's specific protocols that have to be followed. So those are good areas to potentially look to outsource. In terms of academic resources, there's now more than 72 from a directory that exists on this website that I have, screening centers around the world. So these are screening centers in which you can go to, and many of them to do high throughput screening. And then now, and this directory is on the Society for Lab Automation and Screening website that I have here. And now there's a new directory that's up on the same site that's for medicinal chemistry centers. So now, so far, I think there's maybe 6 or 7 that are up there, but it just was initiated, and actually it probably will grow with time as people find out and put their listings on it as well. So if you're looking for screening or looking for chemistry resources, there's a number of centers that offer this. In terms of in vivo pharmacology, one potential source is pharmacy schools. Often you can contact people, and they'll actually work with people outside their institutions in terms of some of the pharmacology. And then animal models. So it was mentioned about some of the foundations that will support animal model development, and so those are actually good resources to go to potentially for advice on people that you could use to do animal models as part of your project. And then there's some CROs that I list here. Psychogenics, JSW Research, SRI, and various academic labs. So these are places that you can look for and talk to as in terms of resources. And then, of course, the internet. I mean, it's just, it always amazes me the amount of resources that you can find from the internet. If you're looking to buy equipment, there's a website called Lab Autopedia, and this has comparisons of equipment. It's not just advertisements. They're actually, people have written sections in terms of how to choose appropriate automation, for example. And they also have various sections written on technologies and tools. There's also this LabMan blog, so you can post something and say I'm looking to do such and such, and you can get responses this way. As well as LinkedIn also runs some discussion groups as well that you can post things and get people's responses. In terms of assay technologies, Eli Lilly a number of years ago wrote an assay guidance manual. Then NIH was given permission to transfer and have a link on their website. And actually, I'm part of a group of editors that are now updating this assay guidance manual. And so this is going to be a Wikipedia kind of format in which you can go up and do searches and look for advice on, like protein-protein interactions, for example, that was mentioned earlier, or what are the options for, kinase type of assays. Also, there's always a lot of web courses and forums. Probably many of you get emails about these kind of things. The Society for Lab Automation and Screening sponsors some of these webinars and short courses. Also, AAAS also has some webinars that you can watch either live or just on your own time as well. And I said, as well as discussion groups. Also, probably your local state biotechnology council. I know in Massachusetts, there's Massachusetts Biotechnology Council. They also sponsor regular programs that give you information or can be resources. So just an example for Lab Autopedia in terms of the automation aspects and genomics tools and technologies, data collection and processing. They'll have conference posters also on their website, as well as recommend certain science books that I have listed here, Life from an RNA World. And there's the website. So these slides actually, as far as I understand, will be available. So the websites and the contacts you'll be able to access. The Assay Guidance Manual really has all types of assays, as I had said. It really has details on strengths and weaknesses, so really give you an understanding of how to make decisions, whether to use peptide substrates, full-length protein substrates, what types of labels are better for high-throughput screening, how to set up various types of assays. And I have the website there as well. Then just in terms of a couple of examples of how high-throughput screening, drug discovery centers, and academics. I just picked first another one at Harvard that I know of, and then I'll talk a little bit about our model. So at the Drug Discovery Center at Harvard, ICCB Longwood, ICCB is Institute for Chemistry and Cell Biology. It's a core facility in which investigators can initiate screening projects with them. They mainly service the Harvard community, but they will work also with people outside of the Harvard community. They have both small molecule libraries as well as RNAi libraries. Collaborators basically come there and perform their own assay development and assays, and they provide all their own reagents. And the staff helps maintain and run the robotics. And there's a certain recharge cost, and that's pretty common. But basically, at the end of the day, you take the compounds you identify or your data from your RNAi screen, and they're yours. So the IP is very simple. They have no claim on it, and then you decide what you do in terms of the next steps. So this is very common as a core type of model. At the laboratory drug discovery that I'm at, it's more collaborative, in which we at the center help drive the drug discovery aspects, and we view it as more of a partnership. It's for the general academic community, both within and outside of Harvard. We have small molecule libraries, and because we're focused mainly on neurodegenerative diseases, our libraries are skewed to be more likely to be blood-brain barrier penetrable, based on physical chemical properties, as has been discussed at this meeting. And the model is that either we or a postdoc from your lab develop an assay, and we run the automation. And we also have medicinal chemistry available to then work on the hits afterwards. In terms of funding, we write joint grants together to get funding for it, and generally there's been joint IP and ownership and joint licensing. We've also actually worked with startup companies as well, and especially virtual startup companies, to do a lot of the biology and the chemistry together. So now the last part, just on what type of contract research organizations exist as an option for resources. So there's fully integrated CROs versus specialists. So the fully integrated CRO will be able to do everything from A to Z, and you can do it all, or you can do just parts of it, versus specialists that may have just a certain aspect, of expertise, like pharmacokinetics or something. So Covance and Recerca are an example of some of the sort of one-stop shopping kind of CROs. And then I've listed Ciprotex, Absorption Systems, Wolf Labs, and Albany Molecular as being more specialists. And actually, you'll hear about Amory after the next speaker. Then there's location. So as probably most of you know, there's been a lot of people going outside to places like India, China, and Europe. And generally, in India and China, a lot of times the prices are lower. But the advantage in the U.S. could be, like we do actually a lot of outsourcing with people just in the Boston area, because the advantage can be that they're very close by and we can have a good interaction with them. And then, of course, you can send out quotes, and you get expensive ones, medium, and low cost vendors. So again, we often rely on word of mouth or experience in terms of various vendors. You can look at within the CRO, you can look at the staff, what the ratio of PhD to MSBS techs versus the years of experience. In some cases, you might want someone who's actually had expertise in your disease area. Could be an advantage in terms of experience that's relevant to your project, and then be able to take advantage of their experience. So in terms of the types of questions you ask them, if you can ascertain whether they have what kind of related projects they've done in the past. So the types of CRO models, for the most part, they're fee-for-service. Basically, the client drives the science, so you basically tell them what you want done. You get a quote, and potentially they can have some input on protocols. For example, for chemistry, they may be able to work out easier chemistry within their scientists. You can get intermediates, chemical intermediates, for example, generated that can help your internal chemistry. And sometimes there can be a per compound rate. So if you just have, a compound or several compounds to make, and they'll give you an estimate for those. But there's also FTE type of rates as well, which I have next. So instead of a fee for service in terms of for X process, how much does it cost, or X compound, or X study, there can be a model where it's really the full-time equivalent. So basically, you pay for one scientist to do the work that you do, or 2 scientists or 3 scientists. So this accounts for some aspects that you don't know how long it's going to take to do certain things. So basically, you're paying for a person to focus on your problem. So what can determine the difference between that is really depends on how much work you have to do. If you just have a specific study, it can be better to do for the fee for service. If you have a fair amount of work, it can be beneficial to do the FTE model. Then there's collaborations, which are less common, in which the CRO may have some shared risk, and so maybe they don't charge the full price, they charge part of the price, or they don't charge at all. But then it's a shared risk model, in which based on licensing or down the road plus side down the road money that may be made. They may then have a part of your company or get some royalties down the road. So that can also happen as well. That is probably a little bit more common in potentially in companies in which they're interested in publications or in marketing, being able to use data that you together you'll generate that can give them more exposure. And as I said, with the collaborations, often it's a reduced fee, some equity, potentially some milestone payments. So the things that we outsource, we do some small amounts of protein purification, but largely we either purchase proteins through outside vendors or use a CRO like Blue Sky to produce proteins that are specifically difficult, maybe difficult or not as good from the ones that are commercially available. Also, as I said, we use CROs for most of our ADME work because our volume just isn't large enough to support, to do it internally, and that includes microsomal stability as well as solubility types of studies, pharmacokinetics. We've been, for example, using absorption systems, profiling. So, if we have a kinase project and we want to do a broad profiling, we've used a couple of different companies basically to look at selectivity. Also, side effect profiling. This gets back to what Frank had mentioned, wanting to put a package together that will be attractive to pharma or biotech. The more you know about your compound in terms of its liabilities, the better the package is. And then mechanistic studies potentially as well. And then supplemental chemistry. So if we have a lot of chemistry going on and not enough chemists, we've done a little bit of overseas chemistry, but we tend to use this one company that's just in Boston called Abergona. And then in vivo efficacy models, we tend to use academic labs. So either it's our collaborator, or in some cases it's another scientist who hadn't been involved in the project that we talked to about testing our compounds. So what my recommendations are is develop a list of CROs with overlapping capabilities. That gives you some flexibility. Often, if you start with just a small pilot project to a CRO before you make more of a commitment, then you get a sense of, what's their turnaround time, what's the actual product, and how you like interacting with them. Some go as far as sending the same project to more than one CRO. If you have the resources, that's fine, but generally we haven't done that. We'll just start working with a new CRO first on something small. Make sure you get updates. I was really impressed when we did a toxicity study with Charles River Labs, for example. The person basically called me every day after each dose and told me how the animals were doing. I mean, that type of interaction, I think, is important. And make sure you get reports that are satisfactory and that you've agreed on ahead of time. So a lot of times when you get quotes from companies, they give you a list of what's included. So make sure you're going to get what you want. And then confidentiality agreements are pretty standard. Also, I think one of the most important things is establishing a relationship with CROs. For example, even though we don't, we're not a big pharma that has a lot of projects that we outsource, but over the years we've established some good relationships with CROs. Quickly, another kind of partnership that people don't often think about is around equipment and technology. So just like Merck was saying, they're often interested in new technology that will make their drug discovery go faster, cheaper, better. What academics can offer to vendors is actually testing new equipment or new technologies in their systems. And often you can get free equipment to try. You can be a demo site and collaborate and basically share the data and publications without actually having to invest right off the bat. And then vendors can offer academics, price discounts, collaborations, and the access to new technology early. So in summary, as you probably have gathered, there are lots of resources out there. Use resources that are outside of your expertise. Talk with others in terms of recommendations. Often try a small project first with a new outsourcing partner. As an anecdote, we tried someone new at one point, and they hadn't really done that study before, but we thought we would give them a chance. And unfortunately, they used some labels for their tubes that they put in the -80 that turned out not to be freezer compliant. So labels fell off samples, and they had no way of really tracing some of the samples. So basically, we were trying to guess, different time points. It was a bit of a disaster. Establish a relationship with them, and make sure you discuss what your expectations are. And I thank you for your attention.
We will hear the mirror presentation that will talk about the drug discovery services perspective on academic collaborations. And Dr. Bruce Molyneux from Albany Molecular Research Incorporated will present.
Thank you, Susanna, and thanks also to Howard for the invitation to speak at the conference here. This is the first conference that I've attended here of the 6 that you've had so far, and I've enjoyed it very much. And I know the intent in giving this conference is for people to learn a lot, which I certainly have over the past couple of days, and it's been very enjoyable. So, as Susanna already mentioned, I'm going to be giving a bit of a mirror presentation, but I'll be perfectly honest with you, I'm going to be saying a little bit more about the contract side of things as well, so there'll be a little bit of overlap with some of the things that Marcy said. So to get it right into it, first of all, I'm a Senior Director of Medicinal Chemistry at Albany Molecular Research, so we're just several hours to the north of New York City. And we're a global leader in drug discovery. We provide valuable resources for doing contract discovery, development, and manufacturing services to advance our clients' goals. So you can see some of the some of the service type offerings and advantages that you have in working with a CRO like AMRI. So first of all, in early drug discovery, we do have an integrated platform that we do use. So we have the in vitro ADMET, also computational chemistry, medicinal chemistry, and in vitro biology that we bring to the game. And these can be used as individual services. They can also be used in an integrated manner to work on a program in a program manner with project leadership. So we have substantial capacity throughout the world. This, as Marcy already alluded to, gives us advantages in being able to offer different prices depending on where you're doing the work in the world. Of course, for customers in the United States, it's easiest to work with us in Albany, and we certainly appreciate the opportunity to do that. Again, I mentioned that a lot of our services are integrated in drug discovery, but we can take you all the way through to actually manufacture of API for commercial use. And as a company our size, we can offer cost-effective solutions for a variety, anything that you want to do, really. So you can see some of the locations here pictured below. So I'm located in the top left there. That's the corporate center up in Albany, New York. So in just a brief overview, what I'd like to discuss today is, what are R&D outsourcing needs of academic laboratories, what drug discovery services are available from CROs, and how do you identify the best CRO to meet your needs, and just a brief slide on outsourcing costs. So AMRI, and I think a lot of big CROs probably have had very limited experience in working with academic investigators over the years. In fact, it was October of last year when I attended a drug discovery and academia conference down in Baltimore, Maryland, where it really opened my eyes to the extent and the breadth of the type of work that was going on. I mean, I was always aware from the literature that, academic investigators are doing really good stuff, but that really brought home to me the magnitude and really the new concerted effort to really do drug discovery, which is really quite refreshing given, where we are, and you see the things that are going on in the U.S. At the big pharma level with reduction of resources, downsizing of staffs, a loss of a lot of brain power, which is basically moving over into Asia, and it's almost like drug discovery in the U.S. has been abandoned. So it's really refreshing to see this groundswell of really good innovative work that's being done in academia. So some of the things that certainly struck me, and these points have been made over the past couple days, is the fact that academic institutions traditionally play a role in identifying novel potential targets, but they really have struggled in terms of being able to push these forward into later stages of the drug discovery process. And that's a quote basically from Jeff Kahn at Vanderbilt. Stephen Fry at University of North Carolina published a survey results last year. And basically, there are approximately 80 academic drug discovery centers, And on average, 50% of the targets that are being investigated are based on unique discoveries that have little validation in the literature. This is something, based on the next quote, drug companies have little really resource at this point to be able to focus on. So it's fantastic that the academic groups have been doing this, and they're very adept at doing this. But the one thing we need to do, and the fourth bullet point obviously, is to do a better job of translation of these early innovations to proof of concept and then on into human clinical trials. So, on what basis can I speak about perspectives on academic discovery? Well, as I just admitted, we've not been working with academic groups all that long, but thanks to the NIH and specifically the NINDS, we were awarded a federal contract to work with the NINDS in collaboration with academic laboratories. So basically, the NINDS identifies academic laboratories with which to work. Then they employ us as medicinal chemists, also our in vitro admit services and computational chemistry to be able to work in concert with them to be able to push things forward. So I want to talk just a little bit based on this experience. And it really, you notice that press clipping was August of last year. So, in fact, we've been working probably for about the last 4 months in collaboration with a lot of the academic groups. So some of this is going to be observations that have been made from that brief period of time, also coupled with 20 years of experience of working with big pharma, biotechs, virtual companies, and on as to how you solve problems in each of these areas. So I start out first with the hit generation, or I think what the NINDS calls exploratory. And here, of course, the prerequisites here are that, of course, you have an idea for a new therapy, which eventually translates into a biological assay that's either target-based or phenotypic-based. You've then validated that screening assay, and then you also have a screening strategy that you've established where you know the assay format that you're working with, and also put some thought into the compounds that you're going to screen to get your first round of hits. So the goal of this stage really is threefold. One, to first do the hit finding to get the actives from your screen, but then there's validation that must occur. And then finally, you have to select which ones that you want to go forward with. Very important decision to make, because this is really going to affect your fate over the next few years if this goes on successfully. If you choose poorly in the hits you take forward, then you're going to have a short-running program. You're not going to go very far. So really what we're trying to do is determine which of the screening hits have the best chance of becoming lead chemical series. So these are some of the important considerations at the validation step. You have to really pay attention to the basics in the hit validation. You have to confirm the bioassay results. You have to make sure that the activity is not due to cytotoxicity. Confirm the integrity of test compounds. Something as simple as making sure that the compounds that you tested were pure and that the chemical structure is what you think it is. I can't tell you the number of times that we've gone through this and we've found out that it wasn't the structure that is the hit, and that really changes the equation. Integral to this process these days is the computational work that can go in here, which can really save you a lot of time and money. Since you're doing in silico screening here, you don't have to do expensive experiments or expensive syntheses in order to move things forward right away. So the computational team at AMRI uses similarity searches, very good chemoinformatic programs that do clustering, virtual screening, structure-based drug design. All these things are brought to bear. And just to give you an example, if you get a hit, one of the first things that our guys do is go in and look at the structure of that molecule, and then they look throughout the rest of the compounds that you've screened to see if there's anything similar that's clustered in the same part of structure space, if you will. You want to know not only what the actives were, but you want to know where some of the hits were that maybe might be a little closer down into the noise of the assay. You can actually learn a lot of early SAR by doing this. and actually pick the better compounds to go forward with. You don't always want to pick the most potent compounds that are going to come out of your screen. You'd much rather have a cluster of compounds that are showing a range of activities to go forward. It gives the medicinal chemist a lot more confidence, the fact that you can do SAR on the actual series. And then, of course, the relevant filters that have been discussed extensively here, and I'm not going to say anything about those, except for don't go crazy with these, straight out of the gun here. Let's not throw the baby away with the bathwater, based on hard numbers in these areas. These are guidelines. Then you want to certainly identify frequent hitters, compounds with reactive functionality, which is a bad place to start from, and often an endpoint in terms of further pursuit of the molecules. Then in terms of the selection process, the emerging SAR, so medicinal chemists will get in there and they'll try to fill in holes in the SAR. So they'll make a few molecules. We're not going to go crazy making hundreds or thousands of compounds at this stage, a few to really see if there is some tractable SAR that we can get within the chemical series. Of course, if you haven't established it already, you want to get cellular proof of concept. And so the functional activity of these molecules is critical and gives you confidence that you've got at least a good starting point for an approach here. And then, of course, the medicinal chemist focuses very keenly on the freedom to operate in chemical space. So if you get into an area that's just beaten to death by hundreds of patents, it may be very tough to maneuver around that chemical space to get novel IP, which is going to be one of the key goals here. Then, of course, synthetic feasibility. Can you run a discovery program? You need to make analogs. You need to make them relatively quickly. You don't want any PhD project syntheses that you have to do to progress compounds. And then, of course, finally, the in vitro admit properties of your molecules. You want to get an early read on what sort of properties you have here. Again, you can get some pretty negative answers here, but it's not necessarily an endpoint. It's not going to kill the program. So what can be outsourced to do this? First of all, in vitro biology surfaces, and Marcy's really spent, I think, did a good job on describing a lot of these things. So I'm not going to say a lot more about this, other than that CROs like Amber, I certainly have really good